Okay, hey, my name is Meg Graham, and today I'm going to talk to you about cellular respiration. Please don't confuse cellular respiration with breathing. With cellular respiration, we're talking about something that occurs at a cellular level. Breathing is what we call mechanical respiration. Now, mechanical respiration is just inhaling and exhaling, but what we're doing is we're exchanging gases. We're primarily trying to bring oxygen in and breathe off CO2. And the reason we do that is because of cellular respiration. Cellular respiration is the process of converting energy that we get in carbohydrates and things like that, like glucose molecules, and we're going to convert that into ATP, which is the form of energy that our cells can use. So cellular respiration is going to require oxygen. That's why we breathe in, but then it creates CO2 or carbon dioxide, and that's a poison to our bodies, so we got to get rid of that, and so we breathe that out. So when you mechanically breathe, you're breathing in oxygen and you're breathing out or exhaling CO2. If you look at ATP, which is the whole purpose of why we're doing cellular respiration, it's producing lots of ATP from storage and energy molecules. If you look at ATP, it's actually called adenosine triphosphate. Well, that tri right there tells you that there's three of something. So there's three phosphate molecules, and each one of these phosphate molecules has a negative charge on it. And if you remember anything about chemical interactions, like charges don't like charges. They repel. So these molecules are just negative, sitting next to negative. They're just like, get me away from this guy. And so you have this really high energy bond among all these phosphates, and they're just bursting with energy. And so our cells can take that energy when we give off a phosphate, and we can use that and do some sort of chemical work or some sort of cellular work. Now, when you take an ATP molecule and you pull off, I don't know what's going on with the pen here, I'm touching too many things. I gotta quit, I'm new at this, sorry. When you take an ATP molecule here and you use one of the phosphates to give off some energy to do something, that leaves you with a molecule called ADP. That D means adenosine diphosphate, which stands for you only have two phosphate molecules now. So anytime you're using these phosphates, you're transferring energy and allowing something to go on within the cell. Now, there's two types of ways that you can make ATP. The first way is called substrate level phosphorylation. And this is where you directly transfer a phosphate molecule and you put it right onto an ADP, and that gives you ATP. So remember, di is 2, so if you add one more, that's 3, so now you have ATP. Okay? The other way that you can make ATP is through a process called oxidative phosphorylation. And this is how you really produce a lot of ATP. This substrate level phosphorylation is not real efficient, and it doesn't happen often, and it makes little amounts of ATP. Okay, but if we want to make big amounts of ATP, that's when we're going to do this oxidative phosphorylation. What we're going to do here is we're going to use energy from something else and kind of push these phosphate molecules onto a ADP to make ATP. So we're still adding a phosphate to an ADP, but it's a different way. Instead of just directly saying, here, you can have this from this, we're going to use some energy from other sources and kind of push a phosphate molecule onto an ADP to make ATP. Now, oxidative, that part of it right there, refers to the fact that this process requires oxygen. Oxygen is going to be used at the very end of this whole series of complicated reactions that we're about to talk to, and it's going to be what we call our final electron acceptor. And so we have to have oxygen to make all this work. All right, let's see what happens. When we're talking about cellular respiration, it's actually divided into 3 different stages. First stage is called glycolysis, and if you look at that word back there, lysis means to break apart. Sorry, I write really sloppy. And then glyco refers to glucose. So we're going to break apart glucose. Well, glucose is a 6-carbon molecule, and what we're going to do is we're going to, there it goes again, we're going to break it into 2 3-carbon molecules. So now we have a 6-carbon molecule broken down into 2 3-carbon molecules. And you're like, well, what's the point of that? Well, every time we break up a molecule, we're releasing energy. And we're going to take that released energy and we're going to convert it to ATP so that now the cell can use it. Because even if I have like if I have a $10 bill, does that mean I can go to the candy machine and buy a candy bar? No, because the candy machine doesn't take $10 bills. I've got the money. I can buy it, but the candy machine won't allow me to. So I've got to go to a change machine and get some quarters and some $1 bills so that I can stick it in there and pay the $1.25 and get my candy bar out of the machine. Now, the cellular respiration works the same way. All it is it's like the change machine of your cells. It turns energy that we have stored in glucose, like we have money in a $10 bill, and it's going to put it into ATP, which would be, in this case, like dollars and quarters, so that we can actually use it and make, get a candy bar or whatever we're going to do in the cell. All right, so this first step, we're going to break apart glucose, and every succeeding step, we're going to be taking energy from that glucose, and we're going to be channeling it so we can make ATP. So our whole goal is to take glucose and then turn it into lots of ATP. All right, so in glycolysis, we break apart glucose into 2,3 carbon molecules, and then we're going to take those 2,3 carbon molecules, and we're going to do this little intermediate step on them, and we're going to make them now 2 carbon molecules. Then we're going to take those 2 carbon molecules, and we're going to go to our next step, which is the citric acid cycle, sometimes known as the Krebs cycle. You see it in literature in different ways. But this is a cycle, which means we're going to start and end with the same thing, but we're going to run these molecules, these 2 carbon molecules, around this cycle, and when we do that, we're going to be throwing off lots of energy in the process all along here. So again, we're still taking this original glucose, and we're going to just keep breaking it down into different things and pulling more energy off. Well, once we run it through the citric acid cycle, the things that we're left with, we're going to now run those pieces through the electron transport chain, and this is where you make really lots of ATP. So this is going to be the final step, but that's where we're going to make the most of our ATP. So let's see how it works. We'll start first with glycolysis. As I said before, we take glucose, which is a 6 carbon molecule, and we're going to break it into these 2 molecules called pyruvate. So we go from 6 carbons to 2 3 carbon molecules, and each one of those 3 carbon molecules is called a pyruvate. All right, when we do this, it doesn't require energy on our part. It's an anaerobic process. An in front of anything, or a in front of any word, means not. So it's not aerobic. Aerobic means it requires oxygen. So this is not aerobic. So this is like a part that this can occur in the cytoplasm. It doesn't require any special organelles. It doesn't require any special gases. It just kind of happens, and we can actually just break glucose down with some enzymes into these 2 3 carbon molecules called pyruvate. Well, when we do all this, we actually are throwing off little bits of energy, and so we're going to yield some ATP by what's called substrate level phosphorylation, where we directly transfer we have an ADP molecule, and we directly transfer a phosphate to it, and that gives you ATP. Okay? Also, what we're going to do is we're going to make some NADH. And you're like, what the heck is NADH? I don't care. Let's talk about it for just a second, because it's important. Okay? NADH stands for nicotinamide adenine dinucleotide. Don't worry about learning that word. Just know NADH. And what you need to know about NADH is that it is an electron carrier that carries our energy. So it's going to pick up an electron, it's going to pick up a hydrogen ion, and when it does that, it's trapping that energy in this molecule of NADH. So NADH is going to be where we've got our energy stored now. So let's go back to where we were. We had 1 6 carbon molecule of glucose. We converted it to 2 3 carbon molecules of pyruvate. And when we did that, we made a little bit of ATP, and we made some NADH. Okay, now we've got energy stored in that NADH, and we're going to come back later and use that. We've already made some ATP here, so that's good, because that's what we're trying to do. So let's move on to our next step then. We have this intermediate step where we take our 3 carbon pyruvate molecule, and we're going to convert it into a 2 carbon molecule called acetyl-CoA. And don't worry about why we're doing this or what all's going on. all I want you to remember is when you go from this pyruvate to acetyl-CoA, you're going to trap some more energy in another NADH. Okay? And so now we've got another NADH that we're going to come back to later on and pull some more energy out of that. All right. So now we're ready with this acetyl-CoA to go to our second step of cellular respiration, which is the Krebs cycle or the citric acid cycle. All right. So we're going to take the energy that we made and stored in acetyl-CoA, or we didn't make it, but we've got it stored in acetyl-CoA, and we're going to turn that into ATP. All right, so what do we need to know? It's considered aerobic, but it's not really, so don't get hung up on that and don't get worried about the details, but just know that it occurs in the mitochondria. Well, that's that specialized little organ in the cell. It's out in the cytoplasm, and we call it the powerhouse of the cell because that's where we really make the energy. So all of this is going to go on inside of the mitochondria, and so it's going to require some special enzymes and some special things to make it happen, but it takes place in that mitochondria. You can't just have this happen anywhere in the cell. And then two, remember, it's a cycle. A cycle means you're going to start with something, and the particular molecule that we're going to start with, I'm going to write OOA. It stands for oxaloacetic acid, and don't worry about knowing it. But that's where we start with the citric acid cycle. And so we're going to add to that our acetyl-CoA. Oops, if I can write. And we're going to put it in this cycle that starts and ends always with oxaloacetic acid. And we're going to run it through the cycle. And when we do this, we're going to go through a series of reactions. And each time we're going to be breaking down this molecule more and more and more to harvest the energy. So we're going to take this 2-carbon acetyl-CoA. We're going to put it with a 4. Whoa, what happened here? Let me go back. Pushing too many buttons. We're going to put it with a 4-carbon oxaloacetic acid molecule that stays in our cycle. Okay, that's just part of the cycle. So now we've got 6 carbons together, and we're going to run them through the cycle. And we're going to break this molecule up piece by piece, and every time we make a change, we're going to throw off energy. So we're going to put some of the energy in an NADH right here, and we're going to throw off a CO2. So we're going to break off one of those carbons, but when we break off that carbon, we're also putting that energy into this NADH molecule. Well, then we're going to do that again and break off another carbon, and that's going to make us put more energy in another NADH molecule. So now we've got some energy stored in these 2 NADH molecules. Then we're going to take a little bit of energy when we start rearranging some of these molecules, and we're going to make a molecule of ATP through that substrate level phosphorylation where we directly transfer a phosphate molecule and make some ATP. Well, that's good. That's what we're trying to do. But we didn't make much. We just made one molecule there. Then we're going to keep going around the cycle, and we're going to throw off a little more energy, and this time we're going to put it in a molecule called FADH2. Now, that stands for, and you can see it down here, flavine adenine dinucleotide. Don't get worried about what it is. It's very similar to nicotinamide, just say NADH, okay? It stores energy. It just doesn't store quite as much energy as NADH. So it stores energy. So we're going to have lots of energy in there, and we're going to come back to that later on. So that's a good thing that we've made. And then we keep going around the cycle, and we're going to throw off one more NADH. So by the time we're all done, we've made 1 molecule of ATP, we've made 3 molecules of NADH, and 1 molecule of FADH2. Well, this is where we want to go. We're making ATP. That's the whole process here. But these 2 things right here, we know they're storing energy because they are grabbing electrons. That's what they do, and that's where the energy is. So now we've got to get the energy out of these 2 molecules and make it turn into ATP. So that leads us to our next step, which is called the electron transport series. Now, the electron transport series, it says series because it's a series of reactions. You take, if you think about like the TVA dams, you block them up and you build this dam and it holds water back. Well, if you were to just bust open the dam, that water would just flow out real quick and destroy everything in its path. Well, we don't want to do that. So what we want to do is we want to take that energy that's stored in that water behind the dam, And instead of letting it flow out all at once, we want to just let it flow out a little bit at a time. Just a little bit here, a little bit there. So you end up with this series of reactions. And TVA does the same thing. They let it run through turbines, and we use it to make electricity. Well, we're going to use the NADH. I don't know why this keeps doing this. Apparently, if I'm hitting something on here too hard, it doesn't like that. So I'm going to write real softly because it doesn't like it when I write hard. So we're going to do the NADH, and we're going to take the FADH2. And remember, we said it didn't store quite as much energy. And we're going to just start letting these go through little bitty reactions called oxidation reduction reactions. And I'm just going to put ox-redux. Okay? Oxidation reduction reactions. Don't worry about what they are if you hadn't had chemistry. Just know that we're going to be moving electrons. Okay? And every time you move an electron, you're moving energy around. And so we're just going to do this series of oxidation reduction reactions here where we're bouncing around electrons, but every time we bounce those electrons around, we're releasing energy. And so what we want to do is capture that energy and turn it into ATP. So as it turns out, for every NADH molecule that you run through this series of reactions, you're going to end up yielding 3 ATP molecules. And for every FADH2 that you run through this series of reactions, you're going to end up making 2 ATPs per molecule. And this is kind of it's an indirect use of the energy. So we take all of the energy that's being released here and we use it to push phosphates onto an ADP, and that makes us have ATP. So that's why you're breaking this down, so you can actually use that energy really easily. And then at the very end here, you have your oxygen, which we said you've got to have oxygen to make cellular respiration. So somebody's got to take those electrons after they've been bounced around through all these different reactions and hang on to them. And so at the end, oxygen is going to take the electron, it's going to take hydrogen ions, it's actually going to take 2, and we're going to turn that into water. So if you look at the whole process of cellular respiration, you've started with glucose, and you've made lots of ATP, and you've made some water. So everything that was in this glucose molecule, we've either converted it to ATP or put some of those leftover molecules in water. And that's the whole process of cellular respiration. It's not that hard. But remember, as we were doing that, we were throwing off carbon molecules in the form of carbon dioxide. And that is a poison for our cells. We've got to get rid of that carbon dioxide, so we have to breathe it out and exhale it, or it will poison our cells. So let's look at a summary here. All right, we start with glycolysis, which is where we take a 6-carbon glucose, and we break it down into 2 3-carbon molecules. Each one of those is called pyruvate. Now we're going to take each one of those pyruvate molecules and we're going to send them through one little intermediate step here, and that's this conversion right here where we make them into 2 carbon molecules of acetyl-CoA. When we do all of these steps, we're throwing off a little bit of ATP here, we're throwing off a little bit of ATP here, and then we're making some NADH. So the main thing we've done is we've made a little ATP, but in the process we've got our energy stored in this NADH. So that's kind of what we're going to keep on hanging on to. Now we take what's left. We have now two carbon molecules of acetyl-CoA, and we're going to send each one of these through the Krebs cycle. So you got this cycle where you got a bunch of reactions here, and you're going to send this one through a cycle too and go through a bunch of reactions here. And in this process, we're going to throw off CO2s, we're going to throw off NADHs, we're going to throw off ATP, we're going to throw off FADH, and that's FADH2. And so now we've got a little bit of energy, but we've got a lot of stored energy here. And so now we've got to take these 2 molecules, FADH and NADH2, and send those to the electron transport series. And when we do that, we just go through this series of oxidation reduction reactions where we do little bitty steps, simple little steps, and we're throwing off a little bit of energy every time, and we take that energy to push a phosphate molecule onto an ADP and make lots of ATP. And then finally we have oxygen that's down here at the end, and it accepts our electron and some hydrogen, and then that turns into water, and we always use water in the cells, so that's no big deal. So in the end, we end up with lots and lots of ATP. Actually, you should have 38 molecules per glucose molecule, and you end up with water, which can just blend in with the cell and not cause any problems. And that's cellular respiration.